Hello everyone, thanks a lot for tuning in. The title of my presentation is Beneath the Ashes, Solve Microbial Diversity After a Burn. And I want to start the presentation by this quote, which is from Leonardo da Vinci and states that we know more about the movement of celestial bodies than about the soil underfoot. And I think this hits the nail on its head by stating that we know so little about soil which is so close to us, and that indicates that how complex the soil microbial diversity is and how little we know and how hard it is to study it. So since soil is so complex, it's really diverse, it's super hard to sequence it and study just a random area of soil. However, one of the ways we can make it a bit easier to study is by burning a plot of land. So what burning does, it reduces the diversity, and then we can take samples at different time points or compare it with unburned samples to see how the diversity is changing. So that's about what we did. We took a restored prairie and a plot of land on a restored prairie. We divided into two parts, burned and unburned. And just to be statistically significant, we took not just burn one plot, we burned 6 of them, and we took samples a week after the burn, 5 months after the burn from the O horizon. So the samples were taken, DNA was extracted and sequenced, it was assembled and then binned into metagenome assembled genomes, and those mags were then dereplicated, and we got 266 medium and high quality metagenome assembled genome. However, one question still remained. Even though we had 266 mags, these were dereplicated, so we didn't know the abundance of each mag in each of the samples. So we combined all the mags in a really big FASTA file and aligned reads from each of the sample on the FASTA file. And that allowed us to calculate the coverage and hence the abundance of each mag, and after abundance, red abundance of each mag in each data set. It gave us a matrix that looks something like this. So each data set and the red abundance of each mag in each data set. And this was actually pretty useful. And one of the things that we did from this matrix was to calculate a PCOA plot of microbial abundance. And as you can see, it's clear from the PCOA plot that the microbial abundance of samples a week after the burn is really different from all the other samples at all the other time points. And if we want to take a closer look, we can see that the PCOA of just the burned and unburned a week after the burn is clearly separate. However, the PCOA of burned and unburned 5 months after the burn merged together. That is, it's not significantly different, and that indicates that the microbial diversity was different a week after the burn, but it's coming back together. It's not the same, but it's coming back together 5 months post-burn. Then the next question is, what are some of the taxa that are increasing? Or what is causing this change after the burn? And I think this slide from the heat map drives home the message well. I know it's a lot to take in, but let's go slowly from top to bottom. We saw a number of different taxa, and each different taxa that are increasing. At the top, we can see it's firmicutes, and firmicutes have been known to increase post-fire because of the spores. So the spores can resist high temperatures and then survive and then replicate when everything else has died. So it's increasing a lot, as you can see on the right on this bar plot. Then we have Pathisi bacteria. Pathisi bacteria is also known as Candida phylum radiata, and this is pretty interesting because these are super reduced phyla. They have extremely reduced genomes. Generally, their genome size is less than 1 megabase pair. So seeing these increase was super surprising, and our hypothesis is that maybe there's a black queen theory is happening where these mags are dependent on other microbial genomes for their survival. Then we have family Micrococcaceae and different Arthobacter genus in them. Those are increasing. Then similarly, we have Blastococcus and Bacillia. All 3 of them have been shown to increase post-fire, but in really different environments. So seeing them increase in our study was super surprising too. And then we have Aeromicrobium. This has also been known to increase in other study, but in our study, out of the 6 mags, 3 were increasing post-fire, while 3 was actually reducing post-fire a week after the burn, which again was interesting. And I'm doing comparative genomic analysis on these MACs to see what's actually causing the decrease or what's causing the increase post-burn. And then if we take a glance at 5 months post-burn, we have all the same taxa. Most of them are all still increasing. However, if you look at the x-axis, the increase has reduced quite a bit compared to a week after the burn. And this again hints at the point that I said earlier, that as we move from a week to 5 months post-burn, the change in microbial or post-burn, the microbial community is coming back more together, and that's why the increase 5 months post-burn is not that much. So currently I'm doing genomics, comparative genomics, to see which MAGs are increasing and why they're increasing, and is there any special traits that we can use to detect what might cause increase in certain MAGs post-fire. And my presentation would be incomplete without giving credits where it's due to these incredible people, and our collaborator, Professor Thea Whitman, who did all the soil sample collection. We are pretty grateful for that, and her constant advice. And feel free to contact me on my email or hit me on LinkedIn. I'm happy to collaborate, talk, etc. Thanks again.